Dear students, today we will talk about histology of the organs of cardiovascular system. Cardiovascular system includes heart and blood vessels. Heart works like pump, it moves blood to the blood vessels, and arteries, they carry blood from the heart to the tissues. Blood goes to the smallest capillaries, which provide gas exchange and exchange of metabolites in the tissues, and then veins collect blood from the capillaries and bring it back to heart. So cardiovascular system provides movement of the blood inside our body, distribution of blood, and also exchange of gases and metabolites inside the tissues. So cardiovascular system provides very important functions. And we'll begin from the histological structure of blood vessels. We have big blood vessels, arteries and veins. They provide macrocirculation, and also we have the smallest blood vessels which provide microcirculation. They include capillaries and another small blood vessels which have diameter less than 100 micrometers, including arterioles, precapillaries, capillaries, postcapillaries, and venules. It's called microcirculation or microcirculatory bed. And arteries and veins, they are big blood vessels, they provide macrocirculation. So we have arteries. From arteries, blood goes to smallest arteries, which are called arterioles. Arterioles carry blood to capillaries. From capillaries, blood goes to venules. Venules, they give their blood to veins. Veins collect blood from venules and bring it back to heart. So here we can see route of circulation in blood vessels. We have two routes of circulation, small and big, and both they have arteries, veins, and between them we have microcirculation or microcirculatory bed. And we will begin from the microcirculatory bed, we will begin from the capillaries, from the smallest blood vessels. So here we can see histological slide which shows us microcirculation, the smallest blood vessels. Here we can see the smallest are the capillaries, and also we can see here arteriole, venule, and they provide circulation inside the tissues, inside the organs. They provide gas exchange, exchange of metabolites, and also waste products. Metabolites and waste products, some of them are released in blood, and it removes, and also nutrients from blood are going to the tissues and organs. So it's necessary for nutrition of the organs, and also capillaries, they provide gas exchange. Oxygen goes from blood to tissues, and carbon dioxide goes from tissues to blood. But another situation we have in lungs. In lungs, they are supported by small routes of circulation. In the lungs, oxygen goes to the blood, and carbon dioxide from blood is released to air in the lungs. So we have opposite kind of gas exchange in lungs. Small blood vessels, they have very thin wall which provides this exchange of metabolites, of gases, of waste products, of nutrients. And let's look at their structure, histological structure of micro blood vessels. Here we can see a structure of capillary, and all blood vessels beginning from capillary till big arteries and veins and even heart, their inner lining is provided by endothelium. Endothelium is special type of epithelium. It's simple squamous epithelium. Here we can see simple squamous cells. And the same cells are in the capillaries, in the arteries, in veins, and even in the heart chambers. So endothelium provides lining, inner lining of all cardiovascular system. So what is specific in the endothelium? Endothelial cells, they form inner lining of the cardiovascular system. They protect underlying layers of blood vessels, and also they provide gas exchange and exchange of metabolites between blood and tissues. That's why endothelial cells are very thin and they have this squamous shape. Here we can see endothelial lining of the blood vessel, and in normal condition it's continuous. In normal condition we haven't interruptions between endothelial cells, and when we have this situation, thrombus may be developed. It will close this place of damage. Here we can see endothelium, endothelial cells, simple squamous cells, boundaries between the cells, and they are squamous in shape. And it's blood vessel on the cross section, and here we can see endothelial lining of blood vessel and endothelial cells. They are nuclei, and cells they have squamous shape, and this squamous shape and thin thickness of cytoplasm provides exchange between blood and underlying tissues. Endothelial cells, they have bases. There is basement membrane, which is characteristic for all kinds of epithelia, and endothelium, it's specific type of epithelium which also has basement membrane. Here we can see endothelium and basement membrane, and those two layers are essential for all blood vessels. Here we can see their structure. And here we can see small capillaries, venule, arterial, and in other blood vessels, and they have endothelial lining. And we can see nuclei of endothelial cells here, and capillaries, they have this thickness of endothelial cells and basement membrane, and some capillaries hasn't any other structures. So typical capillary has endothelial lining of endothelial cells, which is composed of endothelial cells. Also, there is basement membrane. It's essential. And sometimes basement membrane is divided into two layers, and there are gaps between layers. Those gaps contain special cells, which are called pericytes. Here we can see pericyte cell. Those cells are described as cells of connective tissue. They have ability for contractions, and also they may regulate blood flow in the capillary, and also some scientists think that they may participate in the regeneration of the capillaries. So there are pericytes. And also we have additional cells which surround capillaries. They are located outside, not between the layers of basement membrane, not in the gaps of the basement membrane. They are located outside. They are alike pericytes, but they aren't pericyte because they have another location. They are called adventitial cells. Here we can see endothelial cells, pericytes, and adventitial cells. Adventitial cells also, as pericytes, are described as resident cells of connective tissue. They are described as some kind of stem cells. They may participate in the regeneration of blood vessels and of cells of connective tissue. So they follow blood vessels, they follow capillaries, they surround them, and they may participate in the regeneration of connective tissue and blood vessels. So all capillaries, they have three basic types of cells: endothelial cells, which are essential, pericytes, which are located in the gaps of basement membrane, and also we have adventitial cells. And here we can see endothelial cells, endotheliocytes, basement membrane, and also there are cells which surround these capillary cells of connective tissue. Here we can see capillary. It's endothelium in longitudinal section. There are red blood cells, erythrocytes, endothelium, and basement membrane under endothelium. Here we can see endothelium and basement membrane. And there are three different types of the capillaries. This type of capillary is called continuous or somatic type. It has the smallest permeability between all types of the capillaries. It has continuous endothelium, it has continuous basement membrane. And this type is located in brain, it's located in muscles, in skin, so it's somatic type. It's located in those parts of our body where high permeability of the capillaries isn't required. Another type. Here we can see fenestrated type of capillary or visceral type of capillary. Let's compare with previous type. Here we can see endothelium is continuous. In this type, we can see thin regions of cytoplasm of endothelium. Those regions are called fenestra. It's fenestrated type of epithelium of endothelium and fenestrated type of capillary. So this type of capillary has higher permeability than this one, than this type. It's continuous or somatic type, it's fenestrated or visceral type. Visceral type, it means that this type of capillary is located in the inner organs, in viscera. So this type of capillary is located in the inner organs where higher permeability is required compared to this type. There are organs of endocrine system because endocrine cells should release their components, their products via capillary, wall of capillary. For example, thyroid gland has this type of the capillary. Also kidneys, they have this type of capillary, and intestines. Intestinal wall and wall of organs of gastrointestinal tract, they have this type of capillary because absorption takes place there and higher permeability is required. And the third type of capillaries is shown here. Let's compare it to this one. So what has been changed? Here we can see that this type has gaps in the basement membrane. Basement membrane is incomplete, as well as endothelium. There are huge gaps between endothelial cells and gaps inside the endothelial cells. This type of capillary is called sinusoidal type. This type it has the highest permeability between all three types. And where this type of capillary is located? This type is located in those organs where gaps, openings in the wall of capillary are required. This type is located in the red bone marrow and spleen, where formed elements of blood pass through the wall of capillary. Erythrocytes, leukocytes, thrombocytes, they pass through the gaps in the wall of capillary. They may excite from tissues, from the organ to the blood, or they can come back to the tissue of this organ. And also, this type of capillary is present in liver. There are some features of liver structure, and we'll talk about this in the following topics. Why does liver have sinusoidal type of capillaries? So, sinusoidal type of capillary is located in red bone marrow, in spleen, and in the liver. So, generally, we have three types of the capillaries. There are continuous capillaries or sinusoidal or somatic. There are fenestrated capillaries or visceral and sinusoidal capillaries, which have the highest permeability, and the lowest permeability is present in the continuous capillaries. And also continuous capillaries are located in some organs which have barriers between tissues and blood. For example, they are present in brain. There is hematoencephalic barrier between blood and tissue of brain. So here we have three basic types of the capillaries: continuous, fenestrated with fenestra, and sinusoidal, which has the highest permeability. We have capillaries in the microcirculation, but also we have in other types of the blood vessels. We have arterioles, also we have precapillaries, capillaries, postcapillaries, and venules. And also there are arterial venular anastomosis, which provide a direct connection between arterioles and venules. So what is arteriole? Arteriole sometimes is described as the smallest artery, but arterioles, they have specific features. They aren't typical arteries because they haven't only quantitative changes, they also have qualitative changes. They are closer to the capillaries, but also they have special covering. They have smooth myocytes which surround those endothelial tubules, and they form sphincters in the wall of arterioles. So arterioles, they have ability for contractions, and those sphincters, they regulate blood flow in the arterioles. And arterioles, they regulate blood flow in the capillaries. When they are contracted, blood doesn't go to the capillaries, and they keep blood in the arteries, and it causes increase in the arterial pressure. From arterioles, blood goes to the precapillaries, which are very close to arterioles in their histological structure, and both they have transverse striations because of presence of smooth myocytes in their wall. Here we can see capillaries, they haven't transverse striations, but arterioles, they have transverse striations because of presence of smooth myocytes in their wall, and we can see their appearance. Then we have capillaries, the smallest blood vessels, which provide exchange of gases and metabolites. And from the capillaries, blood goes to the postcapillaries, and from postcapillaries, blood goes to venules and then to veins. So postcapillaries and venules, they are very close in their structure to the capillaries. They have almost the same structure, but the main difference is different diameter. Capillaries are small in diameter, venules and postcapillaries are bigger than capillaries. If we will compare arterial and venule, we can see that arterial has usually has transverse striation, and it usually has little bit smaller diameter than corresponding venule of the corresponding order, but it's not the rule. So here we can see that venule, it has only endothelial nuclei, but arterial, it also has transverse striations. And arterial has well-developed smooth myocytes, which provide regulation of blood flow. Here we can see capillary endothelial cells. It's venule. It's very close in structure to capillary, but it has much bigger diameter. And here we can see arteriole. It has additional layer of smooth myocytes which regulate blood flow in the arteriole. And here we can see it's arteriole because it has transverse striations, and it's this one also. It may be branch of arteriole or precapillary. And it's venule. It doesn't have transverse striations, and there are capillaries, the smallest blood vessels. And also we have another extra way of blood flow in the microcirculation. Typical is arteries, arterioles, capillaries, venules, and veins, but we have one more way. It's way via arteriovenular anastomosis. Here we can see extra way. It's extra blood vessel which provides connection between arteriole and postcapillary or venule, between precapillary and postcapillary, or directly between arteriole and venule. In this case, blood goes directly from arteriole to venule without passing through network of the capillaries. Why do we have this type of the anastomosis? This type of circulation, this type of blood vessel regulates blood flow in the microcirculation in the blood vessels. Capillaries may contain more than 10 liters of our blood, but usually we have about 5, 4, 7 liters of blood. So capillaries may contain more blood than we have. And when all sphincters of arterioles will be open, all blood will go to the capillaries and arterial pressure will fall, and it's called collapse. And to prevent this, there are sphincters in the arterioles which regulate blood flow, and there are a lot of empty capillaries. Not all capillaries are filled with blood. There are numerous empty capillaries which are extra, which are needed to provide extra circulation, and only about 20% of capillaries are working. Another are closed. And to provide circulation, there are some extra ways which are arteriovenular anastomosis. Blood goes through this anastomosis and doesn't go to some capillaries which are empty in this case. And when we need, arterioles open their sphincters and blood is distributed in the empty capillaries, and we can see redness, for example, in some region. it happens when do we need this to provide extra blood support, extra blood supply of some region. So arterial venular anastomosis, they provide extra way from arteriole directed to the venule. And they are short blood vessels. They are alike capillaries, and there are two types of arterial venular anastomosis. There are typical and atypical. Typical, they aren't permeable for gases, and arterial blood directly goes to the venule. And in atypical, they have structure of capillary and gas exchange takes place, and mixed blood goes to the venule, not arterial. Here we can see different kinds of sphincters of the arteriovenular anastomosis, which provide regulation of blood flow inside them. So there are typical and atypical, and here we can see special sphincter mechanisms which regulate blood flow in the anastomosis. So we have discussed microcirculation, small blood vessels, arterioles, precapillaries, capillaries, postcapillaries, and venules. And now let's go to big blood vessels, to arteries and veins. They form macrocirculation. They are big blood vessels. So let's look at common features of arteries and veins. They have three layers, and inner lining of arteries and veins is endothelium with basement membrane. Endothelium is the same as in the capillaries, so inner lining is the same. Under the endothelium we have thin layer of loose connective tissue. It's called subendothelial layer. Together they are called tunica intima. It's inner layer of the big blood vessels. So tunica intima, it includes endothelium on the basement membrane and thin layer of connective tissue, or subendothelial layer. It's inner layer of arteries and veins. It's the same in arteries and veins. Next layer is tunica media, middle layer of the wall of the artery and vein. It usually contains smooth muscles, it's tunica media, but it may also contain different elastic fibers, elastic membranes, and it depends on type of artery and vein, and we will discuss this later. It's tunica media, and the outermost layer is composed of loose connective tissue, and it's called tunica adventitia. And here we can see three basic layers of the wall of artery and vein: tunica intima, tunica media, and tunica adventitia, or inner, middle, and outer layer. Inner layer, tunica intima, includes endothelium on basement membrane and subendothelial layer, which is loose connective tissue, and loose connective tissue also is present in tunica adventitia, and between them there is tunica media. Usually, tunica intima and tunica adventitia have the same structure in all blood vessels, but their thickness is a little bit different, depending on diameter of blood vessel. But tunica media may differ in different types of blood vessels, and its structure depends on type and functions of blood vessels. So later we'll look what are the specific features of tunica media in different blood vessels. Here we can see artery, its vein, and both they have tunica intima, its innermost layer, tunica media, its tunica media, and tunica adventitia of artery and vein. So let's begin from arteries. Arteries, they carry blood from heart to the tissues, and they have well developed all three layers. And let's look at layers of the wall of the artery. So artery has three basic layers: tunica intima, endothelium, and basement membrane and subendothelial layer. And next is tunica media. But tunica media in arteries has specific features. First of all, it has elastic membranes. Inner elastic membrane, then we have layer of muscles usually, and outer elastic membrane. Usually we have different number of elastic membranes, but inner and outer are essential. They also always are present. Inner elastic membrane and outer, and between them we may have layer of muscles or different elastic membranes and elastic fibers. So inner elastic membrane and outer elastic membrane and muscles usually are present in the tunica media, but later we will discuss what are the features of Tunica Media. And outer layer is Tunica Adventitia. So Tunica Intima and Tunica Adventitia are typical for all blood vessels. Tunica Intima includes endothelium and subendothelial layer, and Tunica Adventitia is a layer of loose connective tissue. And now let's look at structure and specific features of Tunica Media of different arteries. Its typical structure of artery, here we can see endothelium, then subendothelial layer, and here we can see Tunica Media, two elastic membranes, inner and outer, and Tunica Adventitia. And let's look at membranes, elastic membranes of the artery. Why do we have them? First of all, they maintain the shape of the artery. They maintain artery in open condition. Here we can see opened artery, and its shape is maintained by elastic membranes. And also they are flexible and they are stretchable. And when blood runs through the artery, wall of artery may be stretched, and it's provided by the elastic components, elastic membranes. Why they are wavy on these pictures? Because this artery usually it's post-mortal histological slides, and post-mortem smooth myocytes are contracted, and it results in the wavy structure of the elastic membranes because of contraction of the artery. That's why they are wavy. But in the stretched condition, they will be smooth and they will be straight. So it's general structure of the artery. And here we can see endothelium with thin subendothelial layer. Deeper we can see inner elastic membrane. It's usually well developed, but it's usually developed better than outer elastic membrane. Then we have layer of smooth muscles. Somewhere here we have outer elastic membrane and Tunica Adventitia. Here we can see endothelium, basement membrane, subendothelial layer, its inner elastic membrane, layer of smooth muscles, outer elastic membrane, and Tunica Adventitia. Here we can see also elastic membrane, it's well developed, layer of smooth muscles, and Tunica Adventitia, and outer elastic membrane is almost invisible on these pictures. And here you can see inner outer elastic membranes, everything between them it's Tunica Media, inner covering it's endothelium with subendothelial layer it's Tunica Intima, and outer it's Tunica Adventitia. But it's not the only structure of the arteries. Let's compare this and this picture. Here we can see two opposite types of the arteries. Let's compare. This one has only two elastic membranes, inner and outer. This one has numerous elastic membranes, more than two, inner and outer, and numerous elastic membranes between them. But this type has muscles between elastic membranes. This type hasn't muscles, only elastic membranes are present. So we have two different types of the arteries. This artery is artery of the muscular type. This is elastic type. Two opposite types, muscular and elastic. And later we will see that there is middle type, muscular elastic or elastic muscular type, which has signs of both types of arteries. So this has well-developed muscles and only two elastic membranes. This hasn't any muscles but has numerous elastic membranes. And let's look at features of those arteries and places of their localization. First type, muscular type. Usually the smallest arteries are arteries of muscular type. We have them in the inner organs and they regulate blood flow. They form branches which are continued by arterioles. And they, as well as arterioles, have ability for contraction. They have very well developed muscles and they are contractible. That's why they regulate blood flow in the organs and tissues. When they are contracted, they reduce blood flow in some region. When they are dilated, they increase blood flow in those regions. So they regulate blood flow in small regions which are supported by those arteries, and also they maintain arterial pressure. They don't admit to all blood to go to the capillaries. So those sphincters of the small arteries, they are working and they regulate blood flow in small blood vessels. Here we can see structure of muscular arteries, inner and outer elastic membrane, and between them you can see powerful layer of smooth muscles. They regulate blood flow inside inner organs and before arterioles. So they, together with arterioles, regulate blood flow in the capillaries. Here we can see contraction of muscles, and we can see diameter inner lumen is contracted and then it's dilated. Another type, elastic type of arteries. They have numerous elastic membranes. For example, on this picture we can see aorta, the biggest artery in our body, and aorta usually may have from 40 till 70 elastic membranes in tunica media, but it almost doesn't have smooth myocytes. And that's why it hasn't ability for contraction. Why aorta doesn't have ability for contraction? Because it's very big artery and it brings blood to all organs of our body. All blood from heart goes through aorta. And imagine if aorta will be contracted. It will cause very severe complications. That's why big blood vessels, that's why big arteries, aorta, lung trunk or pulmonary artery and their branches, the biggest branches of aorta and pulmonary artery, they are arteries of elastic type. They haven't ability for contraction, but they have well-developed elastic component, elastic membranes, which provide stretchability of the wall of those blood vessels. Because heart pushes out a lot of blood, and this blood may damage artery, may break artery. And to prevent this, those arteries, they have stretchability. They have very well developed elasticity, and it reduces pulse waves and helps to maintain arterial pressure, systolic arterial pressure on the critical level. Don't be higher than critical level. Here we can see blood vessel. It's artery of elastic type, and there are numerous elastic membranes in the tunica media, but smooth myocytes in this case are absent. Here we can see elastic membranes of the aorta or another elastic artery. And here we can see differences of arterial pressure in different blood vessels, and here we can see the biggest range of arterial pressure is present in aorta, and this elasticity of aorta, it reduces those pulse waves and helps to maintain same arterial pressure and prevents break of the arteries, this elasticity. So when pulse wave goes through the aorta, when some amount of blood has been pushed out by the heart to aorta, wall of aorta is stretched and then it's relaxed, stretched and relaxed. So this elasticity provides normal functioning of big blood vessels, but contractibility isn't required in big blood vessels. So here we can see two opposite types of arteries, muscular and elastic, and also we have muscular elastic type. Let's look at this type. We have two elastic membranes, but also we have additional elastic membranes and smooth myocytes. So it's mixed type between this and this. And arteries with middle diameter, they usually have this type of structure. It's muscular elastic type of arteries. So the smallest arteries are arteries of muscular type. Aorta and branches of aorta are arteries of elastic type. and between them we have muscular elastic type. All arteries with middle diameter, they have this type. When artery has bigger diameter, it has better developed elastic components, and smaller arteries, they have less elastic components but better developed muscles. And here we can see artery of middle diameter. It's muscular elastic artery, and it has well developed both components, muscular and elastic. And now let's go to veins. And first of all, let's compare vein to artery. Veins, they have same layers as arteries. They have tunica intima, which includes endothelium with basement membrane and subendothelial layer, it's loose connective tissue. Also, they have tunica media and tunica adventitia. But let's look what has been changed. In veins, we haven't, here we can see vein, it's endothelium, and subendothelial layer, it's tunica intima, it's tunica media, and tunica adventitia. And let's compare artery and vein. In vein, first of all, we cannot find elastic membranes. Elastic membranes are present only in the arteries. In veins, we haven't them. So tunica media in veins contains only smooth myocytes, and we haven't these elastic components. Another feature is thickness of the wall. Let's compare thickness of different layers. In the artery, we can see thicker tunica media and thinner adventitia, and in the vein, we can see well-developed adventitia but thinner tunica media. So it's basic difference between artery and vein. Here we can see vein, and generally veins of this corresponding order, they have thinner wall than artery but bigger diameter, and arteries, they have bigger thickness, better developed tunica media, and veins, they have thinner tunica media but and generally they have thinner walls than arteries. And arteries, because of presence of elastic membranes, they have wavy contour. Veins, they don't have elastic membranes, that's why they have smooth inner contour of lumen. Veins, they haven't elastic membranes, that's why they are collapsed, because elastic membranes, they maintain round shape of arteries, and usually arteries are oval or round on the cross section in the histological slide, but veins usually are collapsed. That's why veins usually have this shape on the section. Here we can see veins, and arteries. Arteries usually are round on the cross section. Veins are oval, they are collapsed because they haven't elastic membranes. And let's look here. It's artery, round are arteries with wavy contour with well-developed tunica media, and veins are collapsed. And let's look here. Artery has well-developed tunica media and thinner tunica adventitia. In vein, we can see that tunica adventitia is thicker than tunica media. Vein has smooth contour and it's collapsed. And artery is round in shape. So it's how we can distinguish artery and vein. And the last feature is presence of valves in the veins. In arteries we haven't valves, and in veins there are valves which prevent backflow of blood. And let's go to the classification of veins. There are different types of veins, and two basic types are muscular and amuscular. Here we can see that this type is typical. It has well-developed muscles in the tunica media. In this case, we can see that there is tunica intima with subendothelial layer, it's thin tunica adventitia, and middle layer, it hasn't muscles, it also contains loose connective tissue. And borders between those layers aren't clear. That's why we can see that in amuscular veins we have only endothelium and layer of loose connective tissue under endothelium. So it's muscular vein, it's amuscular vein. Where do we have muscular veins? They are present usually in the near brain sinuses of brain, venous sinuses. They are muscular veins because contraction of those veins isn't required. Imagine when veins of brain will be contracted, all blood will still present in brain and it may cause severe complications. So muscles aren't required in brain veins, and also some liver veins, also central veins of liver lobules, they also are amuscular. In those cases they are composed of endothelium only, and subendothelial layer is almost invisible. Here we can see a muscular vein of liver. Muscular veins, they have three types. They have 3 degrees of development of muscles. Here we can see vein with middle development of muscles, with poor development of muscles, and with good development of muscles, or with well-developed muscles. So what is different? In this case, we have very thin layer of muscles in tunica media. In this case, muscular layer is developed better, and in this case we can see that muscles are present in tunica media and even in tunica adventitia, and in adventitia they have longitudinal direction. It's muscular vein with good development of muscles. Where they are present? Muscular vein is with poor development of muscles. They are present mainly in upper part of our body because gravitation helps to move blood. And here we can see, and usually veins of upper part of our body, they don't have valves because gravitation prevents backflow of blood. And here we can see veins with middle development of muscles, and those veins, they may be present in upper part of our body as well as in lower part of our body, and usually they have valves. And veins with good development of muscles, they are present in lower limbs. They are present in lower part of our body, and they have well developed tunica adventitia with muscles, and usually they have valves, well developed valves. So we have discussed blood vessels, and now let's go to structure of heart. Heart has 4 chambers, 2 ventricles and 2 atria, and wall of heart, it has 3 main layers. It's endocardium, myocardium, and epicardium. And also heart is located in heart bag, which is called pericardium. And pericardium has 2 layers. It's serous membranes. Parietal layer, it forms heart bag, and visceral layer is continued and it covers heart, and it's called epicardium. So epicardium also may be described as visceral layer of pericardium. It's serous membrane. And let's discuss structure of heart step by step. So here we can see endocardium, the innermost layer. Under this endocardium we have myocardium, it's endocardium, myocardium, epicardium or visceral layer of pericardium, and parietal layer of pericardium or heart bag. And between them there is cavity, gap of pericardial bag. And let's begin from endocardium. So here we can see endocardium, and this diagram shows us structure of endocardium. So this structure reminds us of muscular vein with poor development of muscles. Here we can see endothelium, thin layer of connective tissue which is like a subendothelial layer. This layer of smooth myocytes reminds us of tunica media of vein, and also we have layer of loose connective tissue which is like tunica adventitia of vein. So endocardium is like muscular vein with poor development of muscles. How do we call those layers in heart? We have those layers of endocardium. It's endothelium, subendothelial layer, muscular layer, and connective tissue layer. Layers which are very similar to layers of muscular vein with poor development of muscles. Next layer is myocardium. Here we can see myocardium. It's muscular layer of heart which provides contractions of heart. And it's composed of cardiac muscular tissue. Look here, this layer of smooth myocytes, it's smooth muscular tissue. And here in myocardium, we have another type of muscular tissue. It's cardiac muscular tissue, and it's composed of cardiac myocytes. Here we can see cardiac myocyte, and it has this cylindric or elongated shape, but they usually are branched. They have branched shape. They have intercalated discs which provide communication between neighboring cardiac myocytes. And cardiac myocytes are attached to each other end-to-end. Here we can see end-to-end connections, and between them there are intercalated discs with desmosomes with gap junctions which provide connection, mechanical adjustment. It's provided by desmosomes and adherent junctions. And gap junctions, they act like electric synapses. They participate in the conduction of wave of excitement. Nervous signal is distributed via gap junctions of neighboring cardiac myocytes. And cardiac myocytes, they form anastomosis and branching structure of the myocardium. Here we can see intercalated discs, and also there are anastomoses of the neighboring cardiac myocytes. Here we can see cardiac myocytes and neighboring rows. They have attachment via anastomosis. Here we can see anastomosis between neighboring rows of cardiac myocytes. So here we can see functional muscular fibers, and they form anastomosis and branching structure of the myocardium. And here we can see this structure. And it provides common contraction of all cardiac myocytes during systole at the same time. And cardiac muscle, it hasn't stem cells. That's why regeneration of cardiac muscle is provided only via formation of connective tissue scar. But regeneration of cardiac myocytes is impossible. Here we can see cardiac muscular tissue. There are intercalated discs, anastomosis between neighboring cardiac myocytes, and anastomosis and branching structure, intercalated discs, and transverse striations inside cytoplasm of cardiac myocytes. And here we can see direction of functional muscular fibers inside the heart muscle, but those functional muscular fibers are composed of distinct cells which are called cardiac myocytes. Here we can see wall of heart, its endocardium and its myocardium, and here we can find specific type of cardiac myocytes which are bigger than usual, and those cardiac myocytes, they are conductive cardiac myocytes which form conductive system of heart. We have typical cardiac myocytes and atypical cardiac myocytes, two basic types. Typical, they provide contraction of heart, and atypical, they are divided into two types, secretory and conductive. Secretory cardiac myocytes, they produce pericardial atrial cardiac natriuretic factor, which participates in the exchange of sodium ions. This factor participates in the endocrine system functioning, and also target cells for this hormone are present in the kidneys. So this factor increases release of sodium via kidneys. It's called atrial natriuretic factor or sodium uretic factor. Another atypical cardiac myocytes are conductive cardiac myocytes, and they provide conduction of wave of depolarization inside the heart, and they form conductive system of heart. And they are of some types. And let's look at conductive system. There are Sinusoidal Node, atrioventricular node, its bundle of His, which has right and left branches, and also there are Purkinje fibers which are located under the endocardium, which distributes the signal under the endocardium. So first node in the conductive system, it's Xenoetel Node. It's located in the right atrium, and it's made up of pacemaker cells. Pacemaker cells, they are first type of conductive cardiac myocytes. They generate nerve impulses with some frequency, and they haven't a lot of myofibrils, but they are small in shape and they generate nerve impulses in Sinusoidal node. From Sinusoidal node, nerve impulse goes to atrioventricular node, AV node, which is located near the septum of heart. This node is made up of second type of conductive cardiac myocytes, which conduct signal from Xenoetel node to bundle of His. But also these cells, they are a little bit bigger than previous. They have more myofibrils, but they also may generate nerve impulses. If they don't receive any impulses from SA node, they generate own impulses and they replace function of SA node. And after this, AV node sends information to third type of cells, conductive cardiac myocytes, third type, which forms bundle of His, branches of bundle of His, and Purkinje fibers. There are third type cells. They have the biggest volume, they have the best developed myofibrils, and they may also generate own impulses if they don't receive information from previous sources, but usually they provide only conduction of the information. SA node usually generates impulses with frequency 80 per minute. If AV node doesn't receive impulses from SA node, it may generate own impulses with lower frequency, about 40 times per minute, and Purkinje cells, Purkinje fibers, if they don't receive information from previous two nodes, for example, after infarction of myocardium, they may generate own impulses with frequency about 20 times per minute. But usually SA node generate nerve impulses which are distributed via all those branches, and they achieve all contractive typical cardiac myocytes. So we have three types of conductive cardiac myocytes. First type is located in the SA node, second in AV node, and third forms bundle of His, right and left branches of bundle of His, and Purkinje fibers. They provide distribution and generation of nerve impulses inside heart. And another type of atypical cardiac myocytes provides production of hormones in the atrium of heart. And here we can see Purkinje fibers, third type of conductive cardiac myocytes. They are located under the endocardium in the heart. And outer layer of heart, it's epicardium. It covers heart and it's serous membrane. And epicardium, it's visceral layer of pericardium. So pericardium, it has parietal and visceral layers. Parietal layer forms heart bag and visceral layer covers heart. Parietal layer of pericardium and visceral layer, they have very similar structure. They have mesocilial lining, it's simple squamous epithelium, and there is thin layer of loose connective tissue under this epithelium. And in epicardium, we usually have well-developed adipose tissue, fat which covers heart, and usually this fat follows blood vessels. And pericardium, it has denser connective tissue which forms this heart bag, and it's needed to provide density of this heart bag. So it's pericardium, mesocilium, simple squamous epithelium, and thin layer of loose connective tissue. And the same structure, almost the same, is present in the epicardium with mesocilium, simple squamous epithelium. So here we can see layers of the heart wall, endocardium, which is very similar to a muscular vein with poor development of muscles, myocardium, epicardium, and pericardium. And valves. Valves of heart are formed by endocardium. It's a fold of endocardium which is covered with endothelium, and in the basis we have a lot of collagen and elastic fibers which maintain the shape of the valve. And here we can see valves of heart. It's endocardium which forms both sides of the heart valves. So we have discussed structure of heart, structure of blood vessels, and now let's go to the structure of lymphatic vessels. You know that we have two kinds of vessels. We have blood vessels which contain blood, but also we have another fluid tissue which is called lymph. It's very similar to blood, but there are some differences, and lymph is located in lymphatic vessels. So let's look at structure of lymphatic vessels. Lymphatic vessels, they include lymphatic capillaries and big lymphatic vessels. Lymphatic capillaries, they are very similar to blood capillaries, but they have blind ends. They begin in the tissues, directly from the tissues. They haven't beginning from the arterioles, so we can compare blood capillaries and lymphatic capillary. And they contain lymph. If you remember, lymph doesn't have erythrocytes. It mainly has lymphocytes and single in other kinds of leukocytes. And also lymph has plasma. Plasma in lymph develops as a result of filtration of tissue fluid. Here we can see that some fluid comes to tissues via arterioles, via capillaries, and goes to the matrix of connective tissue, and then after metabolism some amount of fluid goes back to veins, to venules and veins, but some amount of fluid goes from tissue, it undergoes filtration and goes to lymphatic capillaries. It goes to lymph, it forms plasma of lymph. So lymph uptakes extra amount of water from tissues, and also it uptakes different dangerous substances, different bacteria also they may enter in lymph, and lymph brings these substances, and lymphatic vessels they bring these substances, they bring dirty lymph to the lymphatic nodes which provide filtration, and after that lymphatic vessels carry filtrated lymph to blood vessels to veins, and then in veins blood and lymph are mixed. So what is the structure of lymph capillaries? First of all, first difference with blood capillaries, it's blind beginning. They have bigger diameter than blood capillaries, and also they haven't basement membrane. They have anchoring junctions with neighboring tissues, but basement membrane is absent in lymphatic capillaries. So here we can see their structure. And they carry lymph and bring this lymph to bigger lymphatic vessels. Here we can see villus of the small intestine, and here we can see lacteal lumen, it's lymphatic capillary of the intestine. Here we can see this appearance, and here we can see lymphatic capillary, it's arterial, venous, and lymphatic capillaries are alike typical capillaries, but they don't contain formed elements of blood, erythrocytes. They may contain single lymphocytes or sometimes another kinds of leukocytes. And here we can see filtration and formation of lymphatic capillaries, and they form bigger collecting lymphatic vessels, and they carry efferent lymphatic vessels, they carry lymph to lymphatic nodes, they provide filtration, and efferent lymphatic vessels, they carry this lymph to thoracic duct, and here venous blood is mixed with clear filtrated lymph. And what is the structure of the lymphatic vessel, and what is the difference of lymphatic vessels and blood vessels? Lymphatic vessels are very close in their structure to muscular vein. They also have valves, they also have tunica intima, media, and adventitia. The main difference is structure of tunica media. They usually have two layers of smooth myocytes and they have oblique orientation, and it's the only and basic difference between muscular vein and lymphatic vessel, big lymphatic vessel. So we have discussed structure of small blood vessels, of big blood vessels, lymphatic vessels, and also structure, histology of heart. And students, that's all. Thank you for your attention.